Good morning to all. I thank Professor Gopalakrishnan for introducing the center, and you may appreciate that this initiative is very path-breaking in the area of math and computing. And please take full advantage of the talks that are going to be presented. Clarify your doubts because research involves lots of questions. So with no further ado, let us begin the first plenary session. And we have amongst us Dr. Professor Ramesh Hariharan. So Ramesh, many of you may know, he's the CTO of Stan Life Sciences. So I know Ramesh, but not as much as his colleague Vijay. Vijay tells that Ramesh is the Jekyll and Hyde of the group. He, he doesn't watch TV. He likes to code more. So he is the heart of Stan Life Sciences, and he has been a faculty of the institute in the computer science department. He's currently an adjunct professor there. So he did his BTech from IIT Delhi, computer science. And then he did his PhD from Courant Institute in New York. And then I think he spent his postdoc in Germany. And thereafter he has been in IISc. So when I joined IISc, I knew him through the computer that he built. He had 3 other colleagues who joined together and built this. So those devices were much ahead of time. Computer was much ahead of time. It was actually a mobile device for communication and other things. And I think at that time Vijay was very interested in computational biology, so the group oriented towards computational biology. And Ramesh's forte is fast algorithms. So am I correct? Yes. So fast algorithms. So he has leveraged his expertise in fast algorithms to solve many of the big data problems. So we would like to look forward to hearing. Thank you. Can I look at the presentation? Sorry.
So thank you so much for inviting me, Chirag, Sanchit, and Devnath. My talk is going to be a big picture. I changed the title a little bit. A big picture talk. I used to do algorithms a long time ago, and as I progressed in life, and as the world has now progressed in life, everything has become so empirical that that one starts questioning whether the use of theory in practice is still something that is current or it is a thing of the past. Over a period of time, I've also come to realize that just understanding the big picture on where are the problems and communicating that to, hopefully there's a large number of people here who are interested in, what can they do tomorrow. So communicating where are the problems and where are the interesting areas of activity, that's what I'm going to be focusing on this talk. Of course, I'll give you glimpses of what we do as well in the process. And I guess let me check the time. So, I'm going to focus on genomics data because that's what we work on. Reason number 1. Reason number 2 is there's just a tremendous amount of activity happening in this area. There's increasing accessibility. Costs of sequencing are coming down. The cost to generate 1 gigabase of sequence was about $8 to $10 for a few more months, and then it's going to come down to about $3 or $4. And the expectation, so per billion characters, if you want to sequence, it was $10 earlier. It's going to come down to about $3 to $4, and in the next 3 or 4 years, it's probably be under $1. So that's the progression, which means that human genomes, whole genomes, can be sequenced at about $200 to $50, coming down over the next 3 or 4 years to like $100. And that makes the ability to generate data Um, so there's plenty of data, but it comes with a lot of complexity, and it's not as if sequencing is one thing. As I'll show you, it is many different things that you can use for many different applications. And understanding that plethora of applications and the range of applications is going to be useful. And of course, there are going to be so many open questions that we can throw this tool at, which is what I'm going to dwell upon. I think this number has changed. What is the, you're on the TMT, we have a TMT author here. What's the number? 2.9? 3? 3.1. 3.1. So we have 3.1 billion characters into 2 copies in every one of our cells, right? That's what I mean by DNA. And sequencing the DNA means reading all the characters and getting that out. Your DNA is different from mine, so I need to read your DNA specifically, and since mine is different, I need to read mine specifically, etc., and get this set of characters out, right? That's what DNA sequencing is about. And why do we do this? Just an example case. This is literally from yesterday or 2 days ago. We get a number of patients where something goes wrong in the DNA, and you need to figure out what the cause is. So here is a patient with a whole lot of things you can read on the slide, a lot of mental development and physical development issues. And why does this happen? So when this happens, people have the question of why does it happen? Can I understand what went wrong? Once you understand, there are questions of can you fix it? What can you do about it? Those are bigger questions. But can we at least understand what went wrong? And today, by sequencing the DNA of a person in a standard sort of regular medical setting, when we did this, we found that there were 104 genes that were knocked out in this person. You know, each of us has 2 copies of every gene. 1 copy of 104 genes was knocked out completely. So, point being that nature is always playing these experiments on all of us, where it's constantly changing the DNA and letting loose new individuals with different DNAs, and that's part of nature, the evolutionary process. Occasionally, it goes drastically wrong like this. And until a few years ago, until 10 years ago, it wasn't easy to be able to, in a medical context, be able to do things like this. Today we can, right? Get to the bottom of things like this. So that leads us to the first of the 4 or so key areas that I want to talk about, and I call this the rare variant setting. The rare variant setting is where while you and I share, if you look at the differences between any 2 people, it's about a few million characters that might be different. But most of those differences will also be shared by other people. Like if I have an A at a particular location, there'll be others, and you might have something else, a G, but there'll be several others who also have an A and several others who have a D. So those are called common variants. The variation is there at a particular location, but several people have that variation. And then there are rare variants where I might have an A and very few others might have an A. Most others, almost everybody else might have a G, but maybe 1 in 1,000 or 1 in 10,000 or some things like that might have an A at that location. We call those rare variants. And the reason we look at rare variants differently from common variants is that common variants, since they are in so many different people, they have presumably passed the selection filter, and presumably they can't do something that's terrible. Right? Otherwise, you wouldn't see it in so many different people. Rare variants, since they are localized to a smaller set of people, can have more drastic effects, like what I showed you on the last slide. And so and at a scale of billions of people on Earth, rare, even if it's 1 in 1,000, still means millions of people with problems, right? And so there are a lot of people for whom you need to diagnose what the problem is by sequencing the DNA. It still comes under the category of rare variants, but you can put it in the context of scale. So I'll give you an example. This is a personal example in the sense my mother-in-law in her mid-30s started losing vision. This is how a face would look like to her. And this was about 10 years ago that we did the sequencing, and until then we didn't know what is it, why is it that, and her brother also started losing vision in her 30s, and the question was whether her daughters and my wife would also have a similar prognosis or not. So that's called Stargardt disease, is the name of the disease. And so we sequenced, and I'm going to show you this slide to illustrate the one problem that I want to sort of focus on and the main problem in sequencing and rare disease analysis is we are going to find differences, right? When I sequence a person, I'm going to find that this person has an A where somebody else has a G. What does that A mean? Right? That's the fundamental question. And can we predict what that A means? So let me walk you through this example. So when we sequence this particular, my mother-in-law that is, so we see that there are two differences. One of them, just to help you read this slide, this is what a normal person would look like in a particular small section of the DNA. We have narrowed down to that somehow. This is what a normal person would look like, and in her DNA, what would have been a C in most people had become a T. It was a rare variant. You see it much less than 1 in 1,000 people having this. And the question is, what does the C to T change do? And to an extent, we can predict these things today. You can imagine, right, 3 billion characters in the genome, you change a character and you ask me, what will you survive as a result of this change or not? I will be hard pressed to answer that question in general. But for several locations in the DNA, we can today make an attempt. Some of those are exceptionally simple, because in this case, what happens is that we know that genes have this triplet code, right? 3 characters are read at a time. Those 3 characters are converted into what's called an amino acid. So these 3 are converted into an amino acid. Amino acids together, the sequence of amino acids in dark make a protein. Those proteins are involved in chemical reactions, etc. And that's how the process of life continues. When a C becomes a T, the triplet code just changes from a CGA, which codes for an arginine, it becomes a TGA, which is really a stop, saying that the gene stops there. And so one single character change can make the gene stop in its tracks. And making a gene stop, making a program stop prematurely is not a good thing, right? I mean, because the program hasn't done what it was supposed to do. So this was clearly a bad thing. One character change caused this gene to die. But we have 2 copies of every gene typically, and then one copy doesn't do well, the other kicks in and corrects or compensates for it. So there was something else also that was there. So this is now the second mutation that was there. This is the normal picture again, and this is her picture. So there was a G that became an A. And that didn't do something quite so drastic. That just took the amino acid coded by GGA is what's called, I think, glycine, and then that became a E. So now when one amino acid changes to another, in a situation like this, you can be very confident that the change is very drastic. But in a situation like this, you're a little hard pressed to say, okay, something has changed, but is this really the cause of the problem? Because there'll be several million characters like this in each person's DNA where they're different from the average other person. Right? But this character is sitting inside a gene, it changes the amino acid. Even then, there'll be several, maybe thousands of such characters. Okay? So how do we know this is the cause of the problem? Now, in this particular case, we sort of figured out it's the cause of the problem. I'll come back to how we learn whether this is the cause of the problem later. But suffice it to say that these 2 together gave us enough confidence that the reason that this person is losing vision is because these 2 variants together are crippling both copies of a particular gene. That gene is really the garbage collector in the eye. When light shines on the eye, it leads to a lot of side effects, sort of toxic material gets created. Processing photons is never easy. I guess it knocks out things, and so the chemistry is such that toxic material gets created. Somebody has to clear that out. So there are garbage bags in the eye, and there are clearing mechanisms in the eye to clear out these garbage bags, and all that didn't happen because this is sort of the solid waste management gene in the eye, and when that's unable to clear things, the garbage bags build up over 30-40 years, and over a period of time they start killing the cells. Right? So that was the reason, and then we checked my wife, and she didn't have one of these variants, so we know that she will be fine. So that's a example of what we can use this for. But the point I want to dwell on is, coming from this point here, one character changes to another. How do I know what it does? Because many of these changes make no difference at all. They might make our noses a little longer or a little shorter, etc., etc. Some traits might change, but they may not have a drastic effect. But some of these will have a drastic effect. And that is perhaps one of the big questions for us is how do we predict, like make a change, what it does. And to give you more examples, of these changes can have very strange sorts of effects. On the previous slide, you saw that the amino acid changes, perhaps. That's at least one explainable effect. But there are more complicated effects. So here is a complicated effect. So this is a case of beta thalassemia. A T has, this is the normal sequence, there's a T in most of us, that becomes a G. And what does that do? Now this change is sitting inside a gene, but as you know, genes have coding sequences and they have these things called introns. So these are introns which are technically not used by the gene. Only these portions are used, these so-called exons. And this T to G change is sitting deep inside an intron, so it should really not be changing anything about the gene. But that's where we realize that there are complicated processes happening inside the cell. So the way the cell uses this particular gene, this is the HBB gene. It's a very relevant gene because 25% of us are, 4% of us are carriers. 1 in 25 is a carrier for a mutation in this gene. So there'll be a few carriers in this room. But of course, you'll not get the disease because hopefully by sheer random chance your spouse will not be a carrier. But again, at a billion scale, things happen. So coming back, the way this gene is used is that the cell intends to use just this portion and this portion. So it has to remove this entire piece in between, and it uses sequence cues to remove this entire piece in between. So it says, okay, there's a GT here, so that marks the beginning of the sequence that I must remove, and there's an AG here that marks the end of a sequence that I must remove. Of course, there are plenty of GTs and AGs all in between, so it has to pick the right GT and the right AG, which is the challenge. And the way it picks that is by noticing that in front of this AG there is an A and there's a stretch of which is dense in C's and T's here, so that allows it to sort of get to this AG effectively. And this process is called splicing. And because now what happens in our case is that so where is our variant? It's out here. This T has become a G. Okay, and typically, as I said, the AG marks the end of the part that you need to remove. But now, because of this T becoming a G, a new AG has resulted here. And so instead of going to that AG, you will end up going to this AG. And so you will think that the gene starts from here onwards rather than starting from there onwards. So you'll add 11 more characters to the gene. Adding 11 characters is always a bad thing. If you add multiples of 3, it sort of at least keeps things in, that triplet coding stays in frame. 11 is not a multiple of 3, so the whole gene thereafter gets changed. And that's a drastic change that happens. So these sorts of changes, as you can see, are going to be very hard for us to predict. I change a character somewhere there, does it change splicing, what does it do? Right? So one more example. So this is a case where, what was this? This was a little boy who had what is called Fanconi's anemia, in the sense that the repair mechanisms that are missing, DNA repair mechanisms that are missing, so these people are at heavy risk for cancer. They also have anemia, the number of red cells, etc. is low. So in this case, this is the gene, and then the intron starts here, and this one character G changes to an A. So G is what most normal people would have. This boy had an A there. Now, what it does to the coding, the triplet code in the amino acid, is that it does nothing. It is what is called a synonymous change. A G changing to the A, typically the last character changing in a triplet. In most instances, it doesn't change the amino acid. The amino acid stays the same. However, even though the amino acid stayed the same, the presence of this G close to this point, the junction where the splicing of, the task of removal of this intron has to start from there onwards. If you start tweaking characters very close to that, then the splicing thing goes for a toss. So in this particular case, the gene continued in because this G had become an A. Now this does not always happen if a G becomes an A, it doesn't always happen. So in some cases it happens, some cases it doesn't, and that really illustrates the problem that we have, right? I change a character, what does it do? Does it change splicing? Does it change the code? And there are a few other mechanisms that can also, like we had a stop, a premature stop to the gene. You could have a premature start to a gene, or a start could get compromised. You could have a late start, or the regular end of the program could get compromised, and you could have a late end. All kinds of things can happen. How do we make these predictions? That's our biggest challenge. And imagine, getting thousands of cases in a month. You're trying to look at what is going on, and you've got to do this on an industrial scale. And every person you sequence has like 1 million variants. Even if you restrict yourselves to the genes, which are like 1-2% of the genome, you still have tens of thousands of variants, or after doing all of these filtering, you still have few tens of variants, and for each of those you've got to go and figure out what is really happening. How do you make the prediction? So that's sort of an open problem that we have today, and there are only to give you an instance of why this is important also. This was a case where we found such a variant sitting here. These are all the variants that we could see in literature previously known, and this guy was a completely new thing. Nobody had seen it before. The problem was that this child had died, and her mother was pregnant with the next child. And so I'm telling this just to give you the import of why this prediction is important. Now you have this suspicious mutation, which by all accounts looks suspicious, but it's completely novel, and we can never be sure unless we have some way to predict. And there are two only. Today our arsenal is only, we can do computational predictions, AI, etc., whatever, all of that stuff. But those errors, both on the sensitivity, specificity side, are so large that we cannot trust anything there today. So there is a whole, evolution that has to happen there. Today what we can do is we do look at only two things. Has this variant been reported in literature in somebody who has a problem? So people publish case studies, people run studies, and they publish saying, I took 100 people with this disease and sequenced them, and I found the following list of variants. So, and all that is embedded deep inside supplementary information, some tables, some images. So you've got to go somehow get all of that out in a real live context and bring it to bear on a live context. So that is one trick that's used. Has a patient been reported with this variant? And there are so many variants. I mean, with the 3 billion size genome, there's just so much variation that most variants would not have been even encountered, let alone reported, right? The second trick we use is the counter of that, which is, have normal individuals been reported with this variant? If a normal individual has been reported, then you probably, this is not as important a variant, probably not the causative variant. But then you've got to take into account the fact whether this variant was there in both copies of the DNA or one copy and all of that. But taking all of that into account, if you find this in many normal individuals, you probably say this is not an important variant. Look elsewhere. Yeah. And as I said, there's so many variants to deal with in a given individual that this elimination this way is also useful. This is the positive confirmation, this is the elimination, and both ways are useful. The good thing about the latter is that it's easy to get normal individuals, right? And people are creating these large databases of normal individuals, sequencing them and putting them up. So these databases are much larger than these ones. Going and sequencing people with disease, particularly rare disease, is much harder to do. So you have these large databases building up, and you go and look there and see whether lots of normals have it. But so these are our only two tricks today, very primitive at the moment. These will also grow as the amount of data grows, as the amount of sequencing grows. These tricks will become better. But as data grows, hopefully our prediction capabilities will also become better. And we can answer these questions much better and much less laboriously than we are doing today. We have other problems also, is that the mechanics of sequencing is also complicated in the sense that the entire genome you can't read from one end to the other in one long sequence. It's 3 billion characters, right? So you have to read it in small chunks, what are called reads. A read is typically the most common forms of sequencing. A read is only about 150 in length. And in the more evolved forms of sequencing, a read could be like 10,000 in length. But these are expensive, these are much cheaper, and so for the most part one does this, and occasionally one uses this to sort of fill in information that you might not get using these shorter reads. But the reason why shorter reads are a problem, imagine taking a 3.1 billion length genome, breaking it up, chopping it up, and getting 150 at a time. You've got a big jigsaw puzzle solving problem, and that creates complications with certain types of variation. So not all variation is just that one character changes to another. You can have insertions, deletions, you could have small length deletions, you could have very large length deletions. And I want to show you in a large length deletion what happens, and why this mechanics of sequencing starts getting in the way. So imagine that this sequence as written down is the sequence of a normal person, let's say, and then imagine that in a particular patient this chunk in black is deleted, and let's say it's a pretty large chunk, few thousands or longer in length. Then when you take the genome from the patient, so the patient doesn't have this black stuff, all normal people have it, the particular individual whom we are studying does not have the black thing. So now when we take their genome, which is just the blue part, and you chop it up into pieces, how will those pieces look like? So presumably there will be a piece that comes from here which will have TGA, and then that piece will continue from here onwards CAT, right? And so this is what the piece will look like. Now from this piece, how do we infer that there is a large deletion that's happened? We go and compare this with a normal person's genome, which is this one. And the way we compare is standard string matching. You go and take this string and you search for it against this is called the reference sequence, and wherever this goes and matches, that will tell you what's happening. Now of course this will not go and match anywhere, because there's a TGA here but there's no CAT here, and there's a CAT here but there's no TGA here. So this is not going to go and match anywhere. So with these large deletions, and because of these reads being small, it creates a problem of its own, which means that you've got to do what is called split read matching. You've got to take this read, somehow split it into two. Well, you don't know where to split it into two because you don't know where this black portion is. After all, that's what the whole purpose of sequencing is, to figure out what has really happened in the patient. You don't know that. But somehow if you could split it into two at the right place, then you could take this TGA and search for it, you'll get a match here. Take the CAT and search for it, you'll get a match there. Well, how do you split it at the right place? You don't know the right place. Well, you could try a few different possibilities, and you can try all possibilities if you want, or try some, using a bunch of heuristics you can drive down to that point. And when you do that, you'll see that, and with many such reads are there, then half of those will match here, for each read a particular part might match here and the particular part might match there in the reference, and then you can infer that the entire in between part is deleted, right? So now you've got to get extra algorithms. These have all become standard nowadays. 10 years ago when we started all of this, none of this was in place. We had to get all of these things in place, but so this is the extra thing that one needs to do. Now there is something that confounds, by the way, this case was very interesting in that there were two boys with this deletion, 5,000 characters, and their organs were out of place. The stomach went from the left to the right, the liver went from the right to the left, and all kinds of strange things happened because of this deletion. It's so interesting that we can talk about these things abstractly, and then there's a real manifestation and a real person of the impact of these things. And we have no way of predicting, if you delete these characters, what would eventually happen in the person. We are going the other way. We're getting a person with something is happening, and we are going backwards to the sequence and saying, okay, this is perhaps explains that. But there's a problem that even the split read sequencing is not enough for, because what happens is when you sequence people, you can sequence the entire genome, 3 billion characters, but it gets very expensive. So when you use this in clinical context and want to make this affordable, like run it at 10,000 rupees, we run this at like 13,000 rupees in India today, then you've got to do what is called targeted sequencing, in the sense that you want to take out portions from the DNA and only sequence those, and you have to use your knowledge to say which portions should I pull out. So typically you pull out just the exonic regions of all the genes and sequence those. That's called exome sequencing. Now the problem that arises there is if I've pulled out only the exonic regions and I'm only sequencing those, and imagine that big deletion that you saw on the last slide, that started here and ended up here. Okay? So this is the reference genome which has 3 exons, but in the patient's genome, this particular exon is gone because it from here to here is completely deleted in this person. So he has only 2 exons. So if I sequence the exons alone, I will sequence this exon, I'll sequence this exon. I will not sequence anything here because there's nothing here. So, and I can't do split read analysis because there's nothing coming from here. I'm pulling out the exons and sequencing those. So there are no scraps of paper from the jigsaw puzzle that will come from this part at all. So if I were to sequence the whole genome, then there'll be pieces that come from here, pieces that come from here, and then I can do that split game that I talked about. But if I'm only sequencing the exons and these breakpoints are far into the introns, then I'm not going to be able to do anything like that. So in such cases, how do you go and figure this out as to the fact that this exon has gotten deleted? Well, I mean, the approach that you use is, if this was a normal person, then I would have gotten some reads coming from this exon. Since that exon has gotten deleted, and I'm not seeing any reads from that exon, therefore I can perhaps, since I'm not seeing any reads from that exon, I can perhaps infer that that exon has been deleted. But then it's confounded by the fact that I have 2 copies of every gene, so I have 2 copies of every exon. So I'll still see some reads against that exon coming from the other gene copy, even if one gene copy has that exon deleted. So then it becomes a question of quantification. What is the coverage on this exon, and is that reducing? And when we actually, this is the live data, real data on that person. So when you look at the coverage saying how many genes are on every exon, and you take the ratio with the corresponding number for a normal person, you'll see that the ratio is largely around 1 for most of the exons, but for one particular exon it dips way down, and then you know that this exon has gotten deleted. So you've got to do this quantitative, what is called coverage analysis, look at how many reads are going against a particular region in the patient, divide that by how many went on for a normal person, etc. But then if you have to do that, then statistics comes into play. The standard deviation of these ratios is quite large, and unless you can cover it quite a lot to read the same portion hundreds of times, you don't get such a clean picture. And if you have to do it hundreds of times, again cost goes up, etc. Right? And so you're left with all of these conflicting things that you have to resolve in real life to be able to deliver something like this to patients at relatively low cost. So skip that. The net of all of this rare variant analysis business is that today, when we look at what fraction of the cases that come to us that we can sequence and what fraction of those can we solve and say convincingly that this is the place in the DNA where something changed, and that explains your condition. That hovers around 50%, as you can see. These are various disease categories, and some disease categories may be more successful than the others, but hovers around the 50% mark. And then there's another maybe 5% or so where they formally come under this category called variant of unknown significance, that you have strong suspicion, but you don't have convincing evidence. And then there's another category which is just called variants of unknown significance. This is variant of unknown significance with potential deleterious effect. Variant of unknown significance where you don't know what this is doing. It could be the cause of the problem, it may not be. You don't have enough evidence any which way. With VUSDs, you have some more convincing evidence, but it's not all the way. And there's another 20% or so sitting here. So what happens to the rest, right? I mean, we still, at least 60% of the patients are coming into our doors and spending money on sequencing and getting turned back saying, I can't tell you what's happening, right? So that's an open problem. And worldwide, if you look at the publications, all of this is through a mix of targeted and what is called whole exome sequencing. Whole exome is you only sequence the exons. In targeted, you even go down to, I'll pick the genes that I want to sequence because not all genes are relevant, etc. So a lot of this is because of that, and perhaps if you go if everything had been done on whole exomes or everything had been done on whole genomes, these rates might go up. But the best efforts that we've seen published are something like this, right? You take 64 patients, 33 of them get diagnosed with exomes. You do genomes on those, the remainder, 31 of those, and only 6 of those get diagnosed with genomes. So you're still left with a substantial number, right? About 25 out of 64 who are still going undiagnosed. Okay. So what can we do about those? So that leads me to my first, I mean, I'm sure there are people and research groups here, research problem. Can we dig into this problem? So we have blood samples from thousands of undiagnosed cases. They're all sitting there. We have some panel targeted or perhaps whole exome data on most of those. Can we throw the kitchen sink at it and up the diagnostic rate? I mean, can we do whole genome sequencing? Can we do long reads? Can we do better algorithms? I mean, just needs energy and intelligence to be thrown at this problem to see if there's something lurking behind the scenes that we can use to take this to the next level. Right? So if there are research groups of interest, we'll have to write up some sort of a grant. We can of course provide the blood samples, we can provide the data that we have. We'll have to figure out how to do all of this and get it to the next stage. So this is the first section. Now starting the second section of my talk, I want to talk about common variants. So as I said, common variants are what? These are variants that are present. In fact, this section should be titled Rare plus Common Variant Analysis. We'll see why, but common variants are where you and I differ in a million places. I have a A, you have a G at a particular place. My A is also shared by another, let's say 30% of people on Earth, and your G is shared by the remaining 70% of people on Earth. That's a common variant. Why are common variants important? In most diseases, so as I said, rare variants are rare for a reason, because evolution has selected against them and kept them rare because they have very large effects. They can make or break the human body. Common variants have only largely subtle effects, and so they are not subject as much to selection. So they drift along, and the selection, in the absence of selection, they continue to play a role, a more subtle role in contributing to disease without getting sort of very strongly selected out. Okay? So therefore, most age-related diseases, most complex diseases, and even some early onset diseases like autism, the ASD stands for the autism spectrum, even things like that, autism spectrum disorders, seem to be caused for the most part by a small amount due to rare variation and by a large amount due to common variation. So this pie chart tells you relative proportions. See, if you look at the rare variant proportion, it's only under 10%. Common variant is about 50%. And then there is stuff that is unaccounted for. We don't know whether that is outside your DNA and your heredity and other environmental factors. You know, people talk about vaccines, etc. I mean, who knows what all is there? We don't know much about this. But even something as early onset, something that's early onset, you typically believe will be highly genetic, right? Which it looks like it is. But within that genetic pool, only under 10% is caused by these rare variants. Rare variants are easier to explain because they act in somewhat single-handedly, or at least have large effects single-handedly, complemented by other things which might bring up the rear. But common variants don't act single-handedly at all. They act in like large groups, and it's very hard to pinpoint what does each individual guy do when you have such a multifactorial cause for some disease. Okay. A classic example is height. We believe height is heavily genetic, right? And it should be. So when you start asking what fraction of the variance in height is explained by variance in genetics, meaning if I take 100 people or 1,000 people, I look at their variance, I can measure their heights and I can calculate the sample variance, right? It's a simple formula, calculate the sample variance. Then you go and look at their genome sequences and say, how are those genome sequences varying? Of course, if all of them had the same genome sequence and there's so much variation in height, then obviously genetics has nothing to do with that variation. But the genome sequences also vary. There is some variations there. So if you can quantify the variation in the genome sequence, quantify the variation in height, this of course is easy. This you have to do using some statistical methods. And then say how much of this variation is explained by this. That will give you a measure called the heritability. The heritability is the fraction of variance in height explained by the variance in genetics. And for those interested in statistics, the way this is determined is, let's say you look at a large number of, typically people look at millions of places in the genome, and you try to do some sort of a linear model, say at this place in the genome, let's say there's an A and you give it a weight of 10, and this place in the genome there's a G, you give it a weight of 5. These weights are unknown, you have to figure out these weights. And then you combine all of that effect together and you say what is the best combination of weights that will help predict the height. Obviously, if you have so many locations in the genome, then some linear combination can always match the height. That's the problem with linear regression, right? That if you have more input variables than people that you are predicting, then you will have overfitting. So to prevent that overfitting, you use this various tricks. You use these mixed effect models where these weights are not allowed to vary freely. They have to come from a certain distribution, and you sort of optimize the likelihood of that coming from that distribution, and that constrains these weights to not be so free that you can put whatever value you want to overfit. And when you do all of that, you get some fits, and then the best, like with 1 million such common variants, the best you can explain is 48%, meaning variation in genetics by taking 1 million variants, common variants, explaining the fraction of the variation in height that can be explained by variation in genetics is just 48%. If you take these 1 million common variants and you impute other common variants, so there's this notion of imputation which says that the human genome is inherited in blocks, meaning when I inherit my genome from my parents, I inherit one entire copy of the genome from my mother. My mother has 2 copies, but which one does she give down to me? Neither. She actually makes a mosaic of those 2 copies and gives me. So the mosaic will take one chunk from one of her genomic copies, the next chunk from her second genomic copy, and so on, and then stitch it together into a mosaic and pass that on. So in other words, the genome is inherited in chunks. The mosaic is created in chunks. So if you have a variant here, and if you know these chunk sizes, as time elapses they get smaller and smaller, but the thing about the genome being inherited in chunks is that if you know this variant, you can predict what this variant will be, provided these 2 are close together. If they are far apart, then you can't. But because the genome is inherited in chunks, if I tell you in this particular individual at this location this is the variant, you will be able to make some prediction of 50 characters away, what is the variant that person has, even if you don't know what the character is there. So that is called imputation. So if you impute and make guesses on other common variants that are close by to these common variants, you can up it to about 50%. But then when you take whole genome sequencing data, all the variants which occur in more than 1 in 10,000 people, right, with a frequency of more than 1 in 10,000, you get another 20% of predictability thrown in. So in other words, rare plus common variant analysis, and the reason this happens, we believe, is because while common variants are only giving you 50%, and some of this increase from 50 to 70% may be because the 1 million number is going up to a larger number. We have like several millions of common variants, but any one person will have a few million. But we believe that a large chunk of this gain from 50 to 70% is happening because of several of these very rare variants that are coming in here, and those are contributing to the predictability. So in other words, if we have to predict even something as simple as height, let alone complex diseases like diabetes and etc., which happen over several years and they clearly have a strong nature plus nurture component, so it will be much harder to predict those. But even something as simple as height, which gets set within the first 15 or so years of your life, is heavily genetic. We can only predict, to a tune of meaning the variation in genetics only predicts height, the fraction, well, formally this notion of heritability as defined here is only 70%. Okay. And it requires you to take common variants, a whole truckload of them, millions of them, plus all the rare variants and put them together. Now I don't know what else you can put together. I don't know whether people have tried putting structural variants. Structural variants are these large insertions and deletions. Each of us has many of these, and you'll perhaps hear more about it in the next talk that Vinod will talk about population genomics. There are many other questions on, when you do this analysis, does one clump together variants in a gene, or do you do this at a variant by variant basis? How do you do all of this? So a whole bunch of questions around this theme that are commonly that people are dealing with today. And when they take these techniques where they try to look at lots of common variants and rare variants and predict disease state. So height is, of course, that's a fun problem, but if you have to predict disease state, right? Will somebody get coronary artery disease? We keep hearing in the newspapers, this keeps coming, with alarming regularity. Our morning newspapers will be punctuated by this person completely unexpectedly got a heart attack and passed away, right? I mean, it's happening a bit too regular. Every 2 or 3 months, something or the other that happens. So that's called coronary artery disease. We don't have a really good way of predicting, who would get a heart attack out of the blue. Type 2 diabetes, breast cancer, prostate cancer, all these diseases, we can start using these large numbers of common variants and rare variants. As you can see, our ability to handle such complex predictors and make them work in a clinical setting is much more complex and much not easy to do as compared to rare variants, where you can say, okay, here is one variant that caused the disease. I have full control on that variant. I can predict what is really happening with that variant. Here with millions of variants, who knows, am I getting the right prediction? Am I mispredicting? I'll have to run these experiments on lots and lots of people. So people have done that. So there are these big biobanks that have been created. People have built what are called polygenic risk scores, combining together millions of variants and using those to predict disease state. And then they have run those predictions on these biobanks. These biobanks are broken out into European biobanks, Japanese, South Asian. Unfortunately, South Asian not from India, but South Asian in the UK biobank. The Indian efforts, again, Vinod will probably talk more about it, are not as, that will be one of the points I'll get to. We'd like to do a lot more in India. But many different geographies, many different races. And when you apply these predictions to different races, and you measure what is called an odds ratio by standard deviation. So this is a score. It tells you a high score means you are at risk for disease. Low score means you're lesser at risk for disease. And risk for disease purely on account of what you were born with. We are only measuring your inherited genome. You might be smoking all your life, and those risks we cannot handle. This is only on account of what you were born with, right? So that risk itself, when you look at across populations, as the score increases by, let's say, 1 standard deviation, how much does your risk increase? That's what this tells you. So if you take the entire range of score, and the score has a distribution, you can find its standard deviation. And with every standard deviation increase in the European biobank, it leads to a 1.5 times increased risk for coronary artery disease. So, so if you are in the right around the 1 standard deviation away from the mean of the score, then you have a 1.5 times the risk of getting coronary artery disease as the average person. But that 1.5 times seems to vary across populations. South Asians, in us, the risk seems to be a little, with 1 standard deviation, the risk seems to be a little less. With Africans, it seems to be even lower. Now, we don't think this is fundamental biology. We believe this is because these complex predictors, we've still not figured out how to use them across populations, and that we need to understand this much better. So the point of this is that these complex predictors need a lot more work to go across populations and get them to become robust. And also striking is the fact that South Asians are always closest to Europeans. And we have enough evolutionary history also that tells us that South Asians are, there was a European influx or a Central Asian influx not too long ago, and that was complete mixing at that point before the caste system began, and therefore we are not that different from the Europeans in several things, and that also shows up here. But we are different enough that we still need some level of attention to porting these sort of predictors across. So we are still far away from that. And a lot of these sorts of results have been obtained. You need large sample sizes for this, right? Because you're using so many variants, millions of variants. You better have millions of individuals also, otherwise you're going to get a lot of overfitting. And a classic example of these large, biobanks where people have sequenced hundreds of thousands of individuals is the UK biobank. They were so prescient in their ways. In 2005, they set up this project to just collect blood samples and clinical data and follow up people over 10-15 years. And this has become a treasure trove. And they make this available to everybody for about $9,000 or so. Anybody can get access to this. And this is a treasure trove of information. This is something we have to learn from in India and say, we have a lot more people, we have a lot more disease. We can do something like this at a much grander scale. But we need to have one thing which is missing, which is the ability to think 10 years ahead and do things. 10 years ago I would have said, why are you doing all of this? But today, like, there are thousands of papers being written on this, and this resource is like incredibly useful. Running slow, 10 more minutes, right? Or what? Okay. Okay, so then I'll focus on key things. So these polygenic risk scores are today not ready for prime time. We can't apply them on people, etc., because we still have a lot more to learn. But that's the direction we have to go in, because if we have to predict complex disease, we know that the bulk of the burden is going to lie in these common variants plus rare variant combinations, and we'll have to look at lots and lots of variants. We can't look at one of your variants and make predictions. That we can do only for certain diseases. So now can we take a cue from there? This is my second open problem. There are so many diseases. Coronary artery disease is one. There are so many diseases in an Indian context. We have the largest amount of diabetes. We have the largest amount of diabetic retinopathy, nephropathy, all kinds of things. Largest amount of liver disease, fatty liver disease. How do we create these large pools and large cohorts? And like the UK Biobank has done, which means you need blood samples from those, you need to sequence them, and you need clinical information. You need to know what their clinical history looks like, and you might need to track them over the next 10 years. And if you can do that at a scale of millions, then the amount of information that's going to come from that will allow us to predict these complex diseases much, much better. Right? Today we have the India Genome Project going on. Again, Vinod might talk more about it. That's at a 10,000 scale, very, very subscale compared to where the world is at now, in thinking in terms of millions. There's an added bonus to this, which again, this phenomenon was realized a few years ago, that drug discovery is a very arduous, long process, very inefficient process. You have to pick a gene to target, you have to design a molecule to target that gene, then you have to go through a long process that's for the most part stuck in the clinical world, in the preclinical world where you run on animals, etc. And typically the success rate on that has been 5% from beginning of the project to approval by an authority, regulatory authority. What has been found is that if you have drug programs that target genes which have human genetic evidence, then the efficiency of drug discovery goes up by a factor of 2. So where would human genetic evidence come from? It comes from the experiments that nature is doing broadly. It's creating new individuals with variations constantly, and somewhere hidden in there are all kinds of interesting variations that have this human genetic evidence. So if we create these large databases of individuals, sequence them, then we will get interesting human genetic evidence that can serve as interesting new drug targets that can increase the efficiency of drug discovery by a factor of 2, which means several billions of dollars of savings every year if you look at what is spent by the pharma industry. I want to spend a couple of minutes on cancer genomics. I didn't get to this. In cancer, what happens? So far, everything that we've been talking about is on what we are born with. Now, what we are born with turns out to be a small fraction. As I said, the nurture element of it, any age-related disease is going to be driven by that. And a part of the nurture goes back into our DNA and changes the DNA. You get exposed to various things and that changes your DNA. So your DNA changes with your lifetime. I'm going to just go to the open problems and talk about rather than go through. The DNA changes as you age. Therefore, and some of these changes cause cancer. So cancer cells have their own genomes, their own characteristic variations. Now a lot of work has been done in mapping out. There are these atlases where lots of cancers have been sequenced and mapped out, and so we've got a good understanding of that. What we don't have a good understanding of is, because cancer cells have their own DNA sequences, people have started designing drugs against those those variations. And I'll just give you this picture. For lung cancer, for instance, there are all these different variations, and if you have this variation, this set of drugs apply. If you have this variation, this drug applies. If you have this variation, this drug applies. So the whole pharma drug discovery effort has become now so targeted in some cancers, in lung cancer particularly. It's become really this rich landscape of cancer variations, and how can you design drugs that target those variations. But the problem is that in spite of all of that, cancers develop resistance, and you gain months, maybe the best drugs will gain you a year or two, or on average. Some people have much longer survival. But there are these newer categories of drugs called immunotherapy that modulate your immune system interaction, and those seem to, with the tumor, and that does seem to give very durable long-term responses, but only in 15-20% of people. So the question that we're really asking is, can one create these large cohorts with treatment and outcome information, with DNA and RNA sequencing? We are learning that because now the immune system is involved, the microbiomes get involved, because the bacteria inside trigger. So if you have certain types of bacteria, that will trigger the immune system to make appropriate antibodies, and those will then interact with the tumor. So is there a way to look at all of this from the perspective of increasing the number of people who can benefit from any particular therapy? Let's say immune therapy, only 15-20% of the people respond. Can we up that to 30%, 40%? That will require, again, creating these very large atlases with all of this information. So you can see that big data is pretty much everywhere, right? But we need to generate this data. And now this is something that's tough to do in India, because we don't have treatment, enough people getting treated. If I have to have 1 million people treated with immunotherapy, we have probably thousands of people only who are getting treated with immunotherapy. And globally, there's just such a lot of investment on this. People are largely in academic centers and private sectors creating these huge databases, and it's going to be a treasure trove of information. Let me come to my very last open problem slide. And this section the previous section was called cancer genomics. This section is called non-invasive cancer genomics. So to understand cancers, you have to go into the tumor and sequence it. Getting to the tumor requires getting into your body, doing a biopsy, not often easy. What has happened over the last several years is our realization that in the blood, which is easier to access, you have DNA fragments floating from every one of your cells. Okay? And those cells all turn over at some rate, they die, they let loose their DNA fragments. So your blood is a great window to the DNA that's there underneath in the entire body. And DNA changes with age, and therefore some cells will develop some mutations, other cells will develop other mutations, etc. And not only mutations, there are other patterns and signals that change, methylation, etc., epigenomic signals. So many of these signals are there that you could catch from the blood. Now, what people have shown over the last 3 years is that by catching these signals from the blood, you can actually detect cancer early. So 70% of several cancers can be detected early with very high specificity just by doing a blood draw. We are also working on this problem. Our own data, for example, head and neck cancer, it looks like this. What the score is doesn't matter, but you can see if I take a bunch of head and neck cancer patients and a bunch of normals, the head and neck cancer, and this is from just non-invasive blood draw, all of these cluster well below all of these, and so you get this predictor just simply from a blood draw that can differentiate cancer patients from normal individuals, right? So the open problem is that can one create large cohorts of people where we take a blood draw and sequence their what is called cell-free DNA, cfDNA, and not just people with and without cancer, perhaps people with other diseases, because any sort of disease where cells are dying inside, and several diseases cells will let loose this signature out in the blood. And can we, through these atlases, do all of this sequencing to figure out all of these states and then use that to detect disease early non-invasively? These atlases have barely been started being formed, and this is something that we can do on a large scale. We've already collected about 2,000 samples from various sorts of cancers and are looking at it hard, but we need atlases which are probably hundreds of thousands in size. So much more to be done. So net, thank you for your attention. The message I want you to take away is this is the era of big data in biology. You saw 4 applications where if we create these large data sets, and these large data sets will come with all kinds of challenges, all kinds of algorithmic plus biology and fundamental understanding challenges. But the applications hopefully are very clear to you that we can apply them immediately out in the world and make a real difference. Right? So thank you so much.
Actually, I'm from earth science background, but I was interested to know how much work has been done looking at the microbiome relation with the diseases like cardiovascular disease or diabetes in India. Because I see a significant relationship of microbiome with various diseases.
Yeah, so there are 2 types of relationships, right? One type of relationship is, you find these subtle differences. People with heart disease have some small subtle change, but then people without heart disease, the distributions are highly overlapping with and without heart disease, but good enough to publish a paper because you see a small p-value, but we don't know what to do with it in practice. That's one type of difference, and there are plenty of those with the microbiome. The other type of difference is we have enough control on all the variability that we can use that to make predictions. We want to head in that direction, but that needs much, much larger data sets because the effects, the microbiome varies so much. You eat today something, tomorrow you eat something else, you go to a foreign country, your microbiome will change, you come back, it will change back. There's just so much variation. How do you control for all this variation? The one thing I've realized in all of this data business, it's all about one thing, controlling for variation. There is variation all across the place, nothing is predictive. You have to control for that variation, and that's where big data comes in. We just have to be able to, and well, not only big data, we have to have exquisite control on measurement to be able to control for variation. But a lot more needs to be done. I think there are subtle cues, plenty of subtle cues of the type that you're talking about. Even immunotherapy response, there are cues that people with certain microbiomes respond better, etc. All those cues are there, but we are far from having a predictor that we can apply.
And I also don't know how much work is being done because there's like in other countries, they using microbiome, they get into many types of diseases, and it also gives them a lot of clue, like how this person can change the microbiome with time so that he can get rid of that disease.
Those studies are only happening, so don't take anything for granted. Those studies are just in the process. There are cues, you're right, but these large studies now, the NIH has launched a large food study that they measure food intake of various kinds, measure the microbiome, measure disease state. So we need very large studies for all of these things to be able to get a good understanding of what's going on, and all of that is just starting. So you're going to see a deluge of data over the next several years.
Yeah, thanks for this interesting talk. I just have a question on the head and neck part. Cell-free DNA looks very interesting. So in your data, you have shown that the scores are pretty low as compared to the control. At what stage of the tumor that score comes up? Like we can say it only the tumor onsets, or you can see it even before.
So again, we have some evidence, which is that one way to look at the maturity of the tumor is to do what is called staging, right? Pathologists do the staging. So stage 1, stage 2, stage 3, stage 4. So stage 1 for several tumors, in particular head and neck, stage 1 and stage 2 itself, about 70 to 80% of people will show enough evidence in their cell-free DNA. That will go up to 80-90% in stage 3, stage 4. In other cancers I've seen, in like colon cancer, I've seen studies that show that you could have, you could have detected colon cancer 4 years or 3 years in advance. Now, how do you get that sort of? We need samples from people collected few years before they got cancer. Now, again, it needs only one thing: it needs foresight. So people have to invest blindly in collecting samples with the hope that it will get used tomorrow, which the UK Biobank did, which people in China have done. So there's this colon cancer study from China that came out that 4 years in advance of the person getting cancer, they had stored the blood samples, and now they've gone back and sequenced those. So we just need to do these large scale things to be able to answer those questions. Those are not easy questions to answer.
Thank you.
Imagine heart attacks, right? I'd like a blood sample from somebody a month before his heart attack or her heart attack, so that if I have 100,000 such samples, we can just dredge that for every possible signal and see what could have predicted. But how do I get a sample from somebody a month before a heart attack? That's key. So we just have to keep collecting. It has to be part of a regular program. Every month you collect. Some fraction will get a heart attack at the end. You retrospectively go back and look.
Rare genetic variants, right? When we talk about them, how do we identify the causality at the protein level? And is it an important question to ask that why these rare disease genetic variants, they are not being cleaned up by the normal proteasomal machine?
Why the rare disease variants are not?
The proteins that are expressed as a part of the...
No, DNA should not be cleaned up by anything, otherwise you will not be
No,
alive.
essentially the
RNA you're saying is not cleaned up.
Yeah.
So the RNA is not cleaned up because the RNA is not different enough. If it is different enough, it will get cleaned up. If it's, and for example, if there's a premature truncation, we know that nonsense mediated decay happens and it gets cleaned up. But then you are missing the RNA, and that's going to create a problem. So cleaning up is like throwing out the good. I mean, there's nothing there, right? You need it to survive. First question was what?
How can we infer this?
How do we infer that, right? That's, as I said, today our tricks are very few. Do normal people have it? Are there people with patients with disease who have the variant? And a little bit of biology that we understand to predict, coding changes, splicing changes, but very primitive today. That has to evolve. AlphaFold came up with this ability to predict protein structures, but they still don't have. If I tweak one character in the gene, they don't have the ability to predict the new protein structure. All of that is, and presumably in the works.
Thank you.
So thanks for pointing out all the promises of this technology. I have one kind of devil's advocate question. So what are the fundamental limits of the technology in terms of, one I could think of is the heritability is an upper bound. Explain with genetics, you already communicating to the patient. So do you share these probabilities, and do you also say these are the controlling factors that you rightly pointed out? That with this is your probability, the same mutation.
So two answers. So for the first part, the limitations of inherited genomics is the heritability. But as I said, your DNA changes through your lifetime, and you can also monitor the change through cell-free DNA, and that can, in addition to other things, you can monitor metabolomics, etc., from your blood. That can give you an ongoing window into what is happening. So to an extent, that addresses for certain again, everything will have certain applications and will not be applicable universally for everything, etc. Early cancer detection, eminently it's going to just come about in the next few years. Other diseases perhaps, right? Now what do you do with all the shades of grayness and probabilities? Usually those things don't make their way into the clinic. The clinic is a very, very rough and tough place. You go to an oncologist, I mean, he has 5 patients sitting around him, there are 20 more waiting outside. It is not meant for subtlety. It's meant for extreme black and white thinking, and then you need to reduce things to black and white and operate in that world. So typically we operate with more. That's why the rare variants are much easier to operate with. That's where the clinic is at today. Common variants are for cardiovascular risk are starting to get into early clinical settings, but I see lots of problems there that it will create more unknowns than knowns.
Okay, thanks.
But with rare variants, you can do a lot already. Just to give you, as I said, about 20-25% of individuals are carriers for some rare variant or the other for some debilitating or some fairly serious disease. But they will never realize it, and it may never affect them because the luck of the draw just works out. But it's something possibly it's good to know, because at a billion scale, there are enough people who also get into trouble. There are about 2% of people who have fairly strong cancer risk, 1 to 2%. That is on account of rare variants. There's cardiac risk like that. So those are still things you can put out to patients today, or to normal individuals today, to learn their risk.
Practical question. So what is the good, redundancy now in genome sequencing that one can say?
So genome sequencing is still at 38, but the applications widely differ. So if you're doing whole genome sequencing, 30x is 40x, 30x is decent. If you're doing it in a cancer context, you probably have to do 100x, because when you take cancer cells and sequence, not all of them will be cancerous. They'll be different. Some cancers are heterogeneous. Not all of them. All cells don't have the same DNA. Several normal DNA, several multiple clones. If you're doing it, if you're doing targeted sequencing because you want to save cost, then for reasons that I mentioned, the coverage ratios become important. There, you have to control the variation in those coverage ratios. So you have to go into at least 100x, if not a few 100x. If you're doing cell-free DNA sequencing, you have to go to 10,000x or 50,000x, because the signal is tiny, because you have cell DNA coming from all over the body aggregating, and if you want to just catch that tiny fraction coming from cancer cells. So all these applications have different coverages.
And has these baselines been changing over time, that people realize that this is not sufficient, we should do more deep?
Yeah, so for different applications, we've always realized that each application will need its own coverage. If you do targeted inherited genomes, then you need greater coverage. If you're doing cell-free DNA, you need much greater. If you're doing cancer, you need much. And so all of those, when you build these things in a clinical context, you go through a rigorous process of trying out many different coverages and then validating against things, etc. So those are all part of the regulatory process in a clinical world. But we should think of sequencing as not one application, but a whole collection. Each one will have its own coverage requirements.
Okay, if there are no further questions, let us thank Dr. Ramesh again for his wonderful talk.
Thank you.